direct the Stem Cell Program and Gene Therapy Center at the University of California, Davis Health in Sacramento, California. Our team recently had a great meeting with Megan Leston, who, as you all know, is president of the Krujdasha Research Foundation. And we proposed a stem cell gene therapy approach to deliver the two primary genes in the 5P deleted region, the SEMA5A and delta-catenin. So to study this, we plan to transduce the hematopoietic stem cells found in the bone marrow with a vector that encodes both of those genes under the control of safe promoters. That's what drives the expression of the protein. One which we'll call constitutive, and so that the two proteins would be made throughout the body in any cell that comes from the bone marrow stem cells. And then another cell type specific promoter that would only be expressed in the monocyte-like microglial cells that go into the brain from the hematopoietic stem cells, from the engineered hematopoietic stem cells. Then if successful in the preclinical studies that we're proposing, this approach would potentially use the child's own stem cells mobilized into the bloodstream and collected. We would then take those cells into our clean room facility. It's called a Good Manufacturing Practice Facility. We have a very large one here at UC Davis. We take those cells into our clean room facility, add the corrective genes, and then the child would get a light conditioning before having the cells returned to them. And to return the cells, we inject them into the bloodstream, and they find their way back into the bone marrow, where they would live and make corrected cells for the lifetime of the patient. So I started my career over 30 years ago with Dr. Donald Cohn, working on this same type of approach for an immune disorder that affects children, ADA deficiency or bubble baby syndrome. For that disease of the blood forming system, now over 50 children have been functionally cured. And of course, we're very excited and proud of that. Neural disorders are more of a challenge, but we hope that enough of the corrected cells, hematopoietic stem cells, would travel from the bloodstream into the brain as microglia, that's a specific type of brain cell, and produce a lot of the missing proteins to transfer to the neurons. And that's what our team will be testing. So that's our project. I hope to meet some of you in the future, as we're always very much inspired by the patients and patient advocates. Thank you.